Let's get started. Welcome everybody to our Data Science Fridays seminar. I'm delighted to introduce my colleague John Burns, who will give our seminar today. John is not new to Hawaii, but is a new assistant professor at UH Hilo with a very interesting kind of job description, which is half marine science and half data science. So this is one of these really cool kind of emerging areas where, really pulling together important questions in coral reef ecology and health, but powered by new data science techniques. So really looking forward to your seminar. And John has been a major player in our Ikibai project as well. So it's been great having you on board and working with you on all those projects. Do a little bit of a bio for him. John is an assistant professor of marine science and data science at Hilo, one of 4 new faculty hires to build a data science program there. His research is focused on coral health and disease and has investigated the physiological impacts of disease on affected corals and how coral mortality affects the ecology and biology of coral reef ecosystems. He's really done some groundbreaking work recently in developing innovative techniques to create 3-dimensional maps of coral reefs to accurately measure how natural and human-induced disturbances impact ecosystem function. And the title of his seminar today is 3D Modeling Coral Reefs, How Data Science Helps Us Better Understand Coral Reef Ecosystem. So take it away, John. Delighted to have you.
Thanks, Gwen. I appreciate that introduction. That was great. Thank you. Yes. So as Gwen mentioned, I'm a relatively new assistant professor at Hilo, and it's actually, it was great for me that a position this unique would open up that combined marine science and data science, because I never necessarily planned to kind of straddle those two fields. But as I started studying corals and coral disease, I realized we dramatically needed improved technology in our underwater studies. And the reason why we need that is saltwater and technology tend to not mix well at all. And so it's just simply difficult by nature to implement a lot of new technologies. And one thing I've been quite good at in my career is failing frequently and often. So I was willing to take some risks and chances on attempting to develop some new methods that improved our capacity to not just study coral health and coral communities, but then also predict and model how they might respond to our current changing environment. So the talk today I'm going to give, I just want to first say, I didn't do any of this by myself. Almost all of it was in large collaborative teams, lots of support from UH, like the EPSCoR grant, other grants we've brought in. And so it's a big group effort, and I'm going to give a broad overview of various projects with highlights. So if you have burning questions about details, I tried to put little parentheses for the publications, and I'm more than happy to talk with anyone offline about the nitty-gritty, but I just want to highlight specific advances that we've made really by utilizing data science tools. And so the work I do now is in the lab. I started the Multiscale Environmental Graphical Analysis Lab, the Mega Lab, and so we've got a army of undergraduates here, some watching now, that have been helping us do a lot of this work. So yeah, it's been great to be more established here and share all this research with you. So to start, my interest in coral reefs developed early on because these are just outrageously productive and economically valuable systems. They occupy a small part of the globe, yet house an enormous amount of biodiversity. And anyone, which most of you, if you're in Hawaii, who's jumped in on a coral reef, you're typically captivated right away by the Dr. Seuss-like looking organisms and just the multitude of fish and creatures that leave you wondering what on earth was that. And the fun part sometimes is that we still don't know what on earth that was. And so there's still room for discovery. But that brings the twist to the story, in that in recent decades, they've been facing more local and global stressors than ever before. Just here in Hawaii, we had our first series of repeat coral bleaching events. We're starting to see mass mortality. And like I said, working underwater is hard. And there are a number of coral reefs where we've never even had the chance to put human eyes in the water to see it. And satellites can't penetrate that water. So we're sort of in a strange race against time where we develop tools to protect these systems, to provide ourselves the opportunity to explore and discover within these systems. And so that's where a lot of my work has been dedicated, is trying to learn as much as we can to help protect them so we can sustain our path of discovering more and more from these systems. So how can data science help? Well, science diving really has only been, I mean, someone might call me out on this, but really, in full swing, I would say the past 50 years with, institutions having dedicated dive programs and authorized equipment so we can go down and survey these environments. And so while the scuba technology has changed a lot, we have still primarily, up until the last decade, relied on a lot of two-dimensional tools, specifically a lot of plastic pipe and plastic squares. Now, there's nothing wrong with PVC squares. I love PVC. I use it all the time. And it's great statistically because it defines an experimental unit for us, right? We go on the reef, we say, I put these squares out in random locations, I characterize that system. What it can't do, though, is capture all those invigorating feelings you got when you were in the three-dimensional world of a reef. It's very static and it's a very small snapshot. So I went into structure from motion technology sort of wholeheartedly, realizing that using this tool, we can map large areas of the reef in one shot instead of taking these small little square pictures, and also snap the photos together to create three-dimensional models. And this is just an example from a figure we've been using often from a site that doesn't exist anymore, Wildpie. It's been covered by lava, but it's just showing how you can map at a number of spatial scales because it's a very flexible remote sensing technique. And one thing to point out here is the utility of drawing technologies from other fields. And so to improve my focus in coral biology and ecology, I looked elsewhere. And this remote sensing technique and emerging data science tool was very useful because we can map in this sub box, we mapped, a large area of coastline, and then within that you can three-dimensionally reconstruct smaller areas, and within those areas you can reconstruct reef plots down to colonies. So the point being, and I'll just say this now before I explain the methods, it's extremely flexible. There's no one way to do it. It's a tool that you can apply at whatever spatial scale or focus you wish to better understand an organism. But it hadn't been done underwater. And so that was where there was enormous amount of trial and error. And in the time that when I was starting this early in my PhD, computers were not what they are now, as they're on such an accelerated track of progression. And so this was difficult, sometimes frustrating, and required a lot of troubleshooting and failure. But success came along as well. And why I decided to do this, spend all this time that many folks told me I was wasting on a technology that wouldn't work, was because I knew the 3D complexity mattered. I'd had scientists ask me, as someone studying disease, well, what's the point? What does it tell us if a coral gets sick? What does it mean if a coral dies? Is that even important? And I didn't have a good answer, but I knew it was. And I know it is because fundamentally, just looking at this coral on the left, this Passalopera grandis, you can see the dynamic habitat space right there. There's all types of areas for various organisms to reside, to hide from predators, etc. And so knowing that it's there and knowing from a lot of research, I'm aware that it's the structure, the reef formed by the live corals, that drives diversity and function. And if we lose live corals, we lose that. And because they are so diverse morphologically in their shapes and sizes, we have this array of environmental niche space that allows this small area of the globe to support nearly over a quarter of all marine species. And lastly, why I was certain it mattered is that if we lost that and we lose the system, we're losing it from a bottom-up perspective, right? No corals, no reef, no structure, no fish. So then I had to figure out how to actually quantify and measure this. And so that's where I found structure from motion to be a very useful technique. So it's a photogrammetric technique, meaning it comes from the field of photogrammetry. Where it's unique is it can estimate 3D structure from 2D image sequences. So 3D has been attempted and applied underwater way before I did it. I just want to be clear with that. But it required really complex, clunky, and expensive stereo cameras, because up until SFM, you needed 2 cameras calibrated to be able to reconstruct depth, much like your eyes do as you walk around the world that you live within. And so this technique can take a single lens imagery, process the camera metadata, and figure out, as you photograph a scene from multiple angles, how the scene changes in relation to the optics of the lens, and then reconstruct the positions the camera must have been in. And as you do that, if you scale the scene, you can then create an accurate representation of what you were photographing. And so that's why it's structured from motion, is that you need that single lens to be moving around your scene for it to be able to determine three-dimensionality. And this is something that's actually quite common in nature. If I always use the example, if you're sitting around outside and there's some sparrows bugging you for your crumbs, you'll notice they're always bouncing their heads. And the reason is their eyes are on both sides of their heads, so they don't have stereo vision like we do. And so they're doing something that this technique does. They're taking multiple views of the same scene for their brain to determine depth, to make sure you're not going to try grab them as they go in for your crumbs. And so this was developed, and as the algorithms rapidly progressed about 10 years ago, this matching sequence has become faster and faster and more efficient, and it becomes easier to apply to multiple fields. So as an example with corals, this is just a simplification where, imagine these are 3 pictures taken of this reef scene below. And what the tool will do is identify similarities among the pictures. So you take these images with very high overlap, hoping to get about 80% of overlap. And so as it tracks something using scale and invariant feature transform, so it can identify similar pixels and objects, track them image to image, it's able to then identify these points, take the image metadata, and produce a 3D point for every single matched feature among all your images. And then what this looks like is a final product of your reef scene. And so the algorithms then, as I said, take the metadata, figure out where the cameras must have been, and slowly reconstruct pixel by pixel by pixel until you have a cloud of points, all derived from your images. And then you can use some of these computer vision tools to connect the dots, scale the scene, and create a full 3D mesh like you see here, which gives us this very high resolution and realistic representation of the reef scene that we're studying. And it's very nice because it's a far improved baseline to anything we had before with single images taken at multiple locations throughout a reef. So this is actually a study site in the northwestern Hawaiian Islands, where we've been tracking coral health and disease for the last 9 years, at least that I have. But now we get these plots where we can go down with the high resolution imagery overlaid on the reef model. We can identify specific coral species. We can then annotate these species to see how much area is occupied by each coral, to determine the type of habitat complexity provided by these corals. And most importantly, we can track how this changes through time. And so now we can see deviations associated in habitat structure as corals either grow, succumb to mortality, or are impacted by a disturbance event. And so it has dramatically improved our ability to conduct our research and have a baseline that we use in all of our studies. It's also great because those models allow us to annotate, so we can put multiple data products on top of it and explore spatial relationships between things or among things like water quality, even molecular signals, and the corals that we're studying. I first want to talk about 3D structure. So once I made the models, I realized, well, now I need a way to compute what I'm seeing. So historically, complexity had always been measured as rugosity, where divers would literally, you lay down a chain over the contour of the reef and then compare that chain length to its linear distance. So it's very crude. It works well, but it's very crude. But with this technique, we can take a model like you see here and create or convert it into a raster cell. So each cell of the image has XYZ coordinates. And then we can very quickly and simply quantify elements like surface complexity, so the ratio of 3D to 2D surface area. We can look at slope, which essentially computes a rate of change among these cells. And so you can see these bright red high values are all along the borders of most of the colonies where you have a drop in slope, a high rate of change. We can compute curvature, which is a 4th order polynomial, essentially a slope of the slope. And this is actually really nice Because it's a sophisticated way to see where there's concave and convex surface. Where do we get bends inward? Where do we have holes, essentially, or mounds, and not just be simplifying things as flat or sloped or rough or smooth. And lastly, we can also, because we have a raster, compute it across several resolutions, so we can change the size of each of the cells in that raster file. And we can look at how fractal dimension changes. And so on the right here, you can see 4 different plots. One has very little coral, it's low complexity. On the bottom, we have a very highly complex area. And so we can look at how the complexity based on a fractal dimension calculation changes as we increase the resolution of those cells. And if anyone's into this type of math, you would know that if we make the cells large enough, there will be no structure because it'll just be one giant cell. So we stop it once this decrease is nonlinear and starts to asymptote, and we just cap it there. And then you can look at the slope of that line across resolutions, and the higher the slope, we get and indicates a higher complex habitat. And so I really like this fractal dimension metric because it's similar to rugosity, where we're looking at complexity as sort of a function, but it's encompassing the entire plot and looking at it across multiple resolutions. So it's a very nice, comprehensive way to quantify complexity on a coral reef. So the key findings from this work was, one, we can do it. That was the biggest goal I had when I was first trying to do this in my PhD, was to simply show you can reconstruct coral reefs using photogrammetry. It is possible. And we can also then annotate and identify which species are contributing to complexity, and it can be applied to a wide range of studies to improve our understanding of coral health. So we've used it to look at impacts to habitats after bleaching events, after mortality associated with coral disease, and a range of other studies, and it's actually just being utilized more and more now globally in labs around the world studying coral reefs. And importantly, we see that there are species-specific values. And why I highlight this is because it tells us that not all corals are providing the same structure, which means they're going to have varying levels of importance depending on what you're interested in that ecosystem. So some may provide a specific type of habitat while others provide something else. Knowing that can help us prioritize which ones we need to be concerned about given how they influence other reef organisms. So once we did this, we spent literally years since then wondering, well, which metrics are most important? And at what resolution should you quantify them? And what does it tell you about the reef when you receive a certain signal? Because this has been so minimally applied to reef systems, we wanted to make sure we have a sound understanding of what the data is actually telling us, instead of just putting it out there and saying, look, data, it's amazing. And so this is a study that I'm pretty excited about that we did recently, where we said, okay, let's figure out how these work, and let's see if we can get an improved understanding of the performance of these metrics. So now that there's a number of great 3D animation tools, we actually simulated surfaces. So on a reef, there is a ton of noise. There are organisms growing everywhere. Nothing is simple. Everything is weird, for lack of a better term. So it's hard to extract a singular type of structure. So we made it. We made flat surfaces, linear slope, convex slope, concave slope, walls, and overhanging ledges. Then we went out and modeled individual morphologies of corals. So we have common, very important Hawaiian corals, Montipora capitata, a rice coral, Parietes lobata, a mounding coral, and then 2 branching corals, the cauliflower coral and finger coral. And so we took these specific isolated models, and then we quantified metrics to look for patterns in how they perform and respond. And so some of it makes sense, but some of it was quite interesting. So if we look at surface complexity on the left, and I have the simulated surfaces here above and the coral species below, we see that as we go from what we know to be less complex to what we know to be more complex, there is a distinct pattern in increases in these values. So Montiporids, which are sort of low-lying encrusting corals, they have a lot of morphologies. They're very plastic, but we tend to see them sort of less less largely complex like the branching corals. And so we do see a pattern we would somewhat expect, where we get montiporids, then mounding corals, then branching corals, and it increases. And then we see this pattern again if we looked at fractal dimension. So what this tells us is these metrics are very good at capturing that complexity that matches what we see with our eyes, right? Just visually more complex surface tends to produce visually more complex values. Slope, though, is quite interesting. If we look here, the pattern is not the same. We actually see high values here for the mounding corals, and then the branching corals are somewhat lower than that if we look at the mean values. And something happens similar as well on the simulated surfaces. If you look at walls and ledges, they actually have lower slope values. The reason being is that slope is quantifying rate of change. So you get a really high rate of change at the edges of a wall or a ledge, but as you move down it, the rate of change is not as dramatic. And so the moral of the story there is to realize that not all metrics will reflect the pattern that you expect. And for some, like slope, it's actually more important to look at the range of values than central tendency. So if you only looked at the mean, you might get a confusing result that ledges don't have a lot of slope, but it's really that they have specific areas with extreme values of slope. And so doing this is a thorough way to determine how we should look at and deal with these metrics and interpret what it means when we describe a reef habitat. And there's a similar trend with curvature, where if you look at the simulated surfaces for both plan form and profile curvature, which is measuring concave and convex surface, the mean values are almost all around zero for every single surface. But once you get to the wall and the ledges, we get this dramatic increase in range of curvature. And hopefully this is making sense. I'll come back to all of these values later. So hopefully this is clear. And then if we look at curvature among the coral species, very similar thing. Not really much difference in mean values at all, but as you get to the complex branching corals with lots of interstitial space, lots of concavity and convexity, it's represented in the range. And so here we realize that researchers really need to think about using range when they explore these metrics for certain ones rather than average values. But not for all, right? For surface complexity and fractal, those tend to be okay. And then if we take vector ruggedness, which is a way to look at essentially, as it sounds, ruggedness of the surface. It's a very nice metric. It produces a lot of consistent values. And again, we see a similar trend of increasing mean values in the simulated surfaces, but again, more importantly, a very dramatic increase in the range of values. And so we realize for a metric like this, it's important to look at both the trend of mean values, but also your range. And we look at this one across resolutions, like I was saying, because as you increase your resolution, those differences in mean values start to become more pronounced. And with the corals, if you look at it 1 centimeter, we sort of see what we would expect with increases as we get to branching corals. But as we reduce the resolution of our digital elevation model, those small changes on branching corals become less pronounced. So here's another important lesson, that the resolution at which you quantify structure is very important for the output of your results. So the key findings from this study we published this year is, one, each metric does have a unique ability to capture features from corals or underlying reef topography. In the paper, we go so far as to outline which ones at which resolutions are actually best for capturing reef topography, like spurs and grooves, as well as specific coral topographic structure. So there are, in a sense, correct or best practices to how and why you use these metrics. There's going to be pros and cons to whether you use a digital elevation model or a true 3D surface model. For any of those out there that are really into the 3D, you might be wondering, well, why use a digital elevation model at all? And in the paper, we go to describe how when you measure this across a 3D surface, you get into a lot of debatable questions as to how many vertices you should have, how much you need to decimate the mesh. And so it opens up its own Pandora's box of issues. But all of this is important to think about because, as I said, we as a field of coral ecologists and biologists are somewhat running wild now with this technique. And it's really important to determine what's the right approach and way to interpret the data so we can make comparisons among regions and reefs in different areas and have those be reasonable for how we understand these systems. So things like the resolution and using range versus central tendency are all very important in how you interpret these data. And I'll come back to this because what I've been trying to do for years, which I'm excited to share with you all today, is use these to predict things like the abundance of reef organisms, such as fish. And you can't do that effectively unless you know what these structural metrics are representing. So another area of work that we've been focusing on is looking at these models over time to understand how reefs are changing. And so here we've quantified impacts from disturbance by analyzing these metrics of habitat structure before and after events such as coral bleaching. You can use specific data science tools like cloud compare software that can align the point clouds, like the 3D model I showed you earlier, and then apply specified algorithms to quantify statistically if there's deviations that are meaningful and important in terms of changing that actual habitat structure. And this ultimately lets us do a high resolution analysis of how mortality affects structural complexity. Because like I said, I won't lie, when you first look at it sometimes after a bleaching event, it appears as though all the skeleton is intact the same, right? The only visible difference you see is the loss of live tissue. But we need to know, is that the case or is it different because the live tissue is not there? And so this is just one example site. This is LISIANSKI in the northwestern Hawaiian Islands. These are Montipora corals bleaching in 2014. When we returned in 2015, it was pretty much just all rubble. A year later, we had a macroalgae Buddleia outbreak all over the reef, followed by now primarily Halimeda, but very minimal levels of coral growth. And so again, the structure appears to be there, but when we ran these analyses, we saw significant drops in all values of structural complexity. So this graph is really just quickly, to explain it quickly, it's just showing you we see drops in structural complexity, drops in curvature, et cetera. So complexity is changing. And then using cloud compare tools, we can actually look at specific plots, and everywhere in red are areas of significant change. And this actually shocked me that the whole reef was essentially, as we've been tracking it in future years, it's eroding, it's sloughing down, it's shrinking. And you see that. And you can see it here in the volumetric change. And we actually, for the first time, can make quantified conclusions that nearly all the habitat is affected. And we've lost nearly 1.5 cubic meters of reef area, a volume of that habitat. And this is in just one small study plot. So you can imagine how over a large area, this can dramatically influence the reef scape and the ecosystem processes associated with that. So key findings from this approach is just that everywhere we've done it, we see that loss of live coral significantly alters habitat complexity, even right after they've experienced mortality. So that live tissue is important. And mapping these habitats in 3D dramatically has improved our baseline. And so we've been doing this and receiving grants throughout the Hawaiian archipelago and other locations in the Pacific, because it's just such a drastic shift from us having to use snapshot pictures and try piece together what's happening, where now we can get large baseline areas and see. And in a sense, a picture says 1,000 words, and it's very clear to document the destruction. I am excited. Some of our current, future endeavors is building off the fact that we know camera and computing technology is always going to get better, right? That just never stops. And so we're currently working with several other research groups here at UH and other institutions, and some private partnerships trying to create autonomous vehicles, surface or underwater, that can then collect imagery, and we can do huge spatial scales, so we're no longer diver limited. And one thing I would love to do in the future is just try map entire island coastlines or nearshore reef tracts. And once you do that, we don't need to worry about statistics, because we've mapped the whole thing. And the end results will be even easier to understand. Okay, so this I'm very excited about. This isn't even hot off the press. This is hot in the press. This is currently under review. So I know I should wait, but I couldn't help myself. I want to share some of the results, because like I said, I've been talking about how we're going to be able to do this for so long. It's great to finally have some results. And so we've wanted to predict how these changing habitats alter biodiversity. right? So as we lose coral, as 3D structure changes, what does it mean for fish populations? So I work very closely with the NOAA Papahanaumokuakea Marine National Monument, and we go up on annual expeditions, typically just under 30 days, and we do as many surveys as we can at as many sites as we can from Nihoa all the way up to Kure Atoll. And so we wanted to leverage this and ask if habitat influences fish assemblage structure. To do this, there are about 84 reef sites that we surveyed during the Reef Assessment and Monitoring Program in 2017. So at these sites, divers conducted stationary point count fish surveys, and then over the exact same area, did a 3D reconstruction survey. So we had perfect co-location of 3D habitat data with the fish assemblages they're surveying visually. We then reconstructed all the plots using the technology I showed you. And since the data are co-located, we can start to model how the fish assemblages relate to the various habitat metrics I've been explaining to you in this talk. And we ran a series of Bayesian generalized linear models to identify drivers of these assemblage patterns that we were seeing in multiple trophic classes of fish. And so I'm just gonna show you some of the highlight results where if we look at corallivores, we saw strong, significant responses with vector ruggedness at both 1 and 4 centimeters. So as the habitat becomes more complex at these resolutions, we can see a pretty decent increase in the abundance of corallivore fish. If we look at herbivores, we see something different, and this is what I was excited about. Different fish groups respond to different elements of habitat structure. So here, herbivores, which respond to vector ruggedness at a more coarse resolution of 4 centimeters, and they also respond to vector ruggedness at 1 centimeter when that is matched to curvature ranges. And so that was really interesting to us, that it's not just the ruggedness, but also the concave and convex nature of that surface. And so when there's a lot of concavity with negative curvature values, we see an even stronger signal in herbivore response to that habitat complexity. If we look at piscivores, we see a similar and very positive response to vector ruggedness at 1 centimeter, 4 centimeter, and to curvature range. And so now this should make sense why we use the range of curvature rather than mean values from everything I had shown you before, because that indicates a lot of steep changes like ledges and overhangs. And so we had one reviewer that asked what I thought was a funny question, is like, well, why on earth would piscivores respond to habitat change? And I think I'm gonna write I don't know if I'll actually do it, but I just wanted to respond like, because there's more food to eat, which is probably what's going on. If we've got habitat space for them to be able to hide and sneak up on prey, and there's more prey because there's more fish from the complex habitat, we see this nice positive response of piscivores to habitat complexity. And then we can use multivariate statistics to essentially look at all these relationships in one collective analysis and look for some species-specific connections. And so these are always a little tricky to understand. I'll just walk through it here. The circles you see, the pies, are made up of 5 specific species. So Acantharis triostegus, so we've got various herbivores, Tinochaeta strigosus, wrasses, chromis. So some of the primary fish we see on the reefs. And when you see a larger piece of that pie, of that species color, it indicates that at that specific site, you have more of that species than other fish, right? So each of these large circles made up of these pies are sites that we study. And then you start to see some patterns. So we see sites over here seem to be more similar in their makeup of these blue, green, red pies, indicating they share similarities in the abundance of these fish species. Whereas sites over here tend to have a lot more chromis and thalassoma and wrasses, right? So we see those patterns, and then we can look at the parameters pushing these sites into those locations in multivariate space that's driving these the similarities among those sites. And so what tended to be the most dominant, again, was vector ruggedness at both 1 and 4 resolutions and curvature range. And then the locations themselves, when fitted into this, also, as we would expect, explain similarities among sites, right? So where you are is going to dictate similarity in a fish assemblage, but also how much habitat structure your site has in terms of vector ruggedness and curvature. So again, for us, this was a really enjoyable study to work on, because now we're finally identifying specific metrics at specific resolutions that we know influence groups of fish. And so as the habitat changes, we can model and predict how these fish groups are going to respond and what type of reefs we may see under future climate conditions. Okay, so the last one I want to talk about was a recent study wondering, can digital surveys have the capacity to replace human divers on the reefs themselves? So we've always been wondering, you know, are humans better than machines? The debate has gone on and on at which is superior for various tasks. Computer scientists love to get into this. We've been fascinated with in chess games, putting the world's top leaders up against computers. Obviously, Neo versus the Matrix is one of the greatest situations of human versus machine in our time. But in more real terms, it tends to work out like this, where you have an Amazon warehouse where you try and find where humans do certain tasks better than robots and optimize that situation so the system is as efficient as possible. And so we wanted to try it and see, let's compare human performance to digital analysis performance and look at how each compares. And there are pros and cons to both. So those conventional methods I talked about earlier, an advantage is you're there. Literally, you can look at things up close, you can move around the object, you can spend more time if you see something unique, you can see all elements of the structure. The problem is you're breathing air off of a scuba tank, so you're limited. You only have so much time and you want to achieve the goals of your study. There's also what we like to call, you know, the sensory overload factor, where everyone's IQ drops about 50 points the moment their head goes underwater. So, and you factor in nitrogen narcosis. Sometimes there can be reason to take some discrepancy in what people are bringing back on their data sheets. For the 3D methods, we can have repeated observers. We can collect data in a variety of manners, but it does take more computation time and energy. So what we did as part of a EKVI project with 10 undergraduate students here at UH last year in 2019 was go out and conduct standard visual surveys of corals and coral health, and then afterwards take a three-dimensional survey of that plot. And so we get all the data from the human survey, and then we get a digital representation, which we then had different observers analyze strictly on computers. So just to give you a sense of the comparison, when you're in the field, you might look at an area of reef like this, and you have, I don't know, a matter of really seconds, less than a minute, to do a quick estimate of live coral cover. When you're in the lab, you can spend as much time as you want tracing. So let's just see, anyone want to throw out some guesses as to how much live coral is in this image?
I'd say 20%.
Okay, Gwen comes in quick with 20%. Who else has some guesses out there? The more guesses, the more fun. Sophie can't guess though, that'll be cheating. Anyone else?
I would say 45%.
Okay, we get a 45. Molly Asbury on the chat with 25. So far, Gwen's the low bid on 20. Peter goes 80%. Billy comes in 25. Maria 35. See, we've got a bit of groupthink here. I think everyone's afraid to go too low. Oh, wait, no. Billy revises his 15. Eric tries to get just under the current low bid. And I'll stop it there because Sophie just cheated. She typed in nearly correct answer, which is just about 7.5%. And this was an interesting phenomenon we found over and over and over again. And I do the same thing. We all overestimate with our eyes. And it's probably because that's what we're interested in looking at. We focus on the tissue, we have an inherent bias, and we overestimate. So with the digital analysis, we didn't do that nearly as bad. And so then we applied this to a bunch of different health conditions. So all these codes in this table are various diseases or conditions of reduced health. So bleaching, pigmentation response, tube worm infestation, macroalgae, all the way down to tumors, sedimentation damage, etc. What I want you to notice though is what we did is compare the observations in the field, like we just did in our bidding game, to the digital analysis, and then we simply look at the number of times they agree, or the number of times they disagree, or again agree. So either both said a disease was present, one said it was present, the other did not, vice versa, or they both say the disease wasn't present. The reason why we did it this way is because there's no gold standard. We don't know which method is best. And so we did a blinded analysis. So nobody analyzed the same plot that they would have observed in the field. And we had 3 observers blindly analyzed digitally, and then we took the majority rule, so we were as least biased as possible. We matched 191 coral colonies from all the surveys from both digital and in situ assessments, and then we compared these prevalence values to see how they perform. So what we're really comparing here is sensitivity, true positives, versus specificity, true negatives. And this was Dr. Grady Weyenberg, who's also over here at Hilo. He came before me in the data science higher cluster, and he was the mastermind behind this technique. As a statistical wizard, he was aware of this Hui and Walter model that's a latent class model designed for, as I said, diagnostic tests with no gold standard. So we don't assume one is better. We just compare how they perform. And then what's great about a Bayesian approach is we can apply priors. So my work with coral disease over the last 10 years here in Hawaii, along with collaborators, has been summarized in several papers. Jamie Caldwell did a great one in 2016. So it's just a database of all expected prevalence values of coral disease. So we don't have to operate in the blind. We can fit priors to these disease states because we have certain expectations of what the prevalence values should be. And this helps us make sure that none of the models are throwing out something that would be wildly impossible that we would never expect to encounter on the reefs. And if all of this is making you feel like this, don't worry. The nice part about Bayesian stats is when it's all done, because then you get to ask relatively simple questions and receive nice, simple answers like this. The in situ approach had higher sensitivity. It's simple as that. No p-values, no fuss, no muss. You're 7% more likely to detect a health condition if you do it in the field. But the digital method has higher specificity. So you're about a 1.5% less likely to have a false negative if you do it digitally, presumably because you've got more time, right? You have that ability to sit there, look at it, and be really careful in your analysis. And you're probably more likely to detect it in the field because, like I said, you're able to look closely, look around, actually have your eyes right there. Even though those 3D models are high resolution, they don't capture everything perfectly. It's still not your eyes on the reef. And this is nice because I still like to dive for work, and it's good to know that I can do something possibly better than my computer. And then we can break this up into a hierarchical model, so we can look at each disease condition independently, right? So damage, sedimentation, all the way down to bleaching, and we compare the in situ method in red versus the digital in green. And so then we can see certain nuances, like growth anomaly had the biggest discrepancy between the two methods, likely because this is a growth phenomenon. So it's hard to see in an image. It's much easier if you can kind of look around it because it's subtle changes in morphology. Pigmentation response is the same, both methods, because it's a blatant coloration shift. And that's actually picked up very well, if not better, in the images. And for both, bleaching had the highest sensitivity rates, meaning they had the least conflicts between either method because it's, again, easy to detect. And so, like I said, we're doing this not to necessarily really say one is better than the other, but optimize how we use human observers and the computers. So maybe we can allow human divers to spend more time on diseases that are difficult to detect digitally, and that lets them have a more thorough investigation of that, while the others can be quickly annotated back in the lab. And soon that will be made even easier with machine learning tools that can auto detect things like a white tissue bleaching or a strange discoloration. And lastly, we fit this into a curve that no one, at least we're pretty sure, we haven't found anyone that has done this before. So we thought this is fun, right? It's the first time anyone's made a graph like this, where we look at probability on the y-axis and sensitivity difference on the x. So we can actually see, because we took a Bayesian approach, what is the probability of a difference between the two methods. And I've just got single and majority rule to show you the difference, but the point is just noticing the pattern of the curve. Because what that tells us is there's about a 50% chance that the difference exceeds 8%. So that's right around that 7.5% difference, right? So if you ask me, how different is a diver than someone analyzing an image? It's about a 50-50 chance that you could have an 8% difference. There's only a 33% chance, though, that that difference it will exceed 10%, and only a 7% chance that the difference exceeds 15. So how I interpret that is while there is clearly an advantage for certain conditions to observe it in the field, there's still a strong reduction in probability that that difference is going to be anything extremely large. So the digital methods are capable of detecting these conditions and can hopefully help us optimize our approach to studying coral health. So big takeaways here is just that these data science tools are really transforming our monitoring by allowing us to reconstruct these habitats in 3D, have these permanent baselines, and then annotate an array of data onto those models. What I didn't show today just for time is we also do this with molecular data sets as well, and even disease states, physiochemical data, and we can annotate it all over those models and then look for spatial clustering patterns to improve our understanding of the reefscape. And we have a really exciting EPSCoR grant in the works right now that hopefully gets funded that'll allow us to collect samples, map microbiomes back onto the reefscape and see where we find certain microbes and molecules. And we've done that on a small scale, and I would love to do it at larger scales. Dissecting the meaning and methodology of these metrics is something that was very tedious, but we felt like we had to do it in order to have confidence in how we interpret those metrics and really hopefully progress the field to having standardized data that can be comparable to do what we did where we understand how habitat influences biodiversity. That is going to be very important for the future. The reefs are changing, and we need to know how those changes will affect fish. Once we know which corals and which fish are at risk, we have the chance of creating better targeted conservation strategies. And as I've mentioned several times, technology is improving exponentially. And so we're at a time where we really have to keep looking forward and taking risks, trying to implement these new tools. Because if we want a chance at saving these systems, we need to work as fast as at integrating new technologies as they're experiencing environmental change. So we need to take risks, we need to try new things. That's all I've got for you today. I just want to acknowledge a number of people. There's more than I have time to do so, but very much so Dr. Atsuko Fukunaga, my close collaborator at NOAA, as well as Randy Kosaki, who he really makes all the magic happening by pulling the strings on these complex expeditions. My Mega Lab technician, Kylie Pascoe, she actually does all the work. I just get to present it. Brian Hawk, Jason Leonard, Kale Lope, Steve Matadobra, Courtney and Tom at NOAA, all of them. I mean, like I said, this is a huge team effort. My mentor, Dr. Ruth Gates, Dr. Misaki, and Donna all helped me by encouraging me to actually do this when, like I said, everyone told me it was a total fool's errand to try and do something new. And then just other folks I've worked with and all the great students we have in the Mega Lab and the volunteers, as well as a number of funding sources. So with that, yeah, and just again, a huge thanks to EPSCoR and Gwen and EKVI for again letting me continue to pursue my interests, as weird as they seem at some points, because sometimes we have some success amidst the failures. And so with that, I think we have a little bit of time for questions. This is my email, but you can find it on both the lab website and my personal website. And so while we've got questions, I'll keep the URLs up there, but just play some of the 360 video that we collect from our research sites. So if you ever want to see what these sites look like in the monument, we do follow the mission of bringing the monument to the people. And so we host a lot of these videos online on the Coral Health Atlas. So yeah, if you want to break from your Zoom and feel like you're underwater, those are available for you. And thank you to everyone for coming. It's really great to, even though we can't have seminars in person, it's awesome we can all get together one way.
Wow, awesome. Thank you, John. That was great. Boy, it's so fun to see things underwater too. I'm sure we have lots of questions for, we can either put them in the chat, or if I can see you raise your hand, raise your hand. Who has a question for John? Alex does. Alex, go ahead.
I have one technical and one sort of biological that, after chatting with Ruth many years ago, we were thinking about. So one is how does the focal length of the lens affect the calculations and the resolution and things like that? You know, and would you be better off with a pinhole versus a lens or something like that? And the second question I have is there like an optimal crenulation for coral health, you know, a bumpiness, you know, because I know that the coral larvae need a certain environment to stick in. And, you know, if there is something optimal, is there sort of a cascade point where these larvae just don't stick and your coral health declines? What's the
Yeah,
question?
this is good. I feel like I'm defending all over again. Those were great questions. I don't know. For the first one, yeah, it has everything to do with optics. So lucky for me, you know, I was really into photography when I was younger. My uncle is a photographer. And so I in high school, you know, we had, it was still film. And so film is a really great way to understand optics, like your aperture and your shutter speed and film speed, your ISO. And so there is like like about focal length about 24 millimeters is really good, but it's very nuanced. Like you ultimately want the best lens optics with the minimal distortion. So fixed lenses, the better lens quality, the better camera sensor quality, everything just gets better. And so that was like a tricky point training people originally, because a lot of folks, especially doing, you know, a lot like high amounts of surveys, everybody wants the like plug and chug method. You're like, oh, can't I just put it on auto and go? And it's like, no, you need to know what your optics are, because it alters your end product. And then you have to be really careful with the aperture and shutter, because if your aperture, you know, is too large, you start to get too much field of view and blur. So you got to find the happy medium that still lets enough light in due to the attenuation. So it is this fun game of like taking every key parameter from photography and then trying to optimize it each dive. So I do have set guidelines I can give to people, but in reality, when I'm down there, I always try make it as perfectly set to those conditions with light and turbidity. To your second question, I love that idea. I mean, yes, I think there are absolutely minute structural variations that will probably promote things like settlement as well as healthy tissue. But one thing that hopefully you and I get to follow up at some point is I think there's a lot of signaling that goes on that we're just unaware of as well. And so one thing that I get very concerned about with the loss of live corals is how much are those live corals signaling settlement around them? Because we're working hard to very painstakingly annotate every coral in our plots, because no one has really still gotten a handle on how spatially clustered are these organisms. And when we decipher that, we might see things that could lead us to looking for clues in cell signaling that tells corals, look, this is a good place to settle. We know it kind of happens with CCA, but it's not extremely well characterized. So yeah, I would love to take it down to that level.
Okay, we've got two questions in the chat. Let's, Lauren Weiss, do you want to ask your question?
It's already typed out. I think you've got it.
Okay, I will okay, so Lauren's question is, in measuring pigmentation response, both the in situ and digital imaging techniques had a sensitivity of around 60%. Is white light or another active illumination tool used with either technique to compensate for the loss of red light underwater?
Yeah, so, you know, that's funny because that's where the camera can actually be better. You know, your eyes can't deal with shifting attenuation, so you see things on the reef, you know, very muted in terms of true color. The cameras have gotten so good now compared to what I started doing with this that when you shoot, so if you shoot in RAW, I shoot in RAW primarily, and then if you white balance off those sensors, you can get very good true color representation. And so that's why sometimes I feel like it's almost easier to capture pigmentation response in the imagery, because the true color can come out better than what you're able to see with your naked eye.
Awesome. Okay, next question is from Bastian Benlage, our colleague in Guam. His question is, John, could you briefly touch upon what the steps would be to implement automated image annotation methods? The rate limiting step in a lot of image-based survey techniques seem to be the time it takes to annotate the images. Considering the increasing frequency of reefs change, it seems that developing automated and rapid assessment technologies would be very useful. Yeah.
Yeah, it's an excellent question, and you're spot on. The biggest bottleneck is annotation. So the quick answer is there's always a shortcut. The one that works best is annotating by points. So like CoralNet, for instance, is a point annotation tool. So you throw 100 or 1,000 or 10,000 points over your model or your mosaic image. And then based on the pixel characteristics, you can train it using machine learning tools to guess what species is there. And that's actually really does well. It's fairly accurate, performs very well. And so you can use that to annotate and then extrapolate things like coral cover, diversity, species composition, et cetera. But it's still extrapolating. It's not doing everything. And so the true bottleneck is that semantic segmentation, actually annotating every border of every colony. That is just exceptionally hard because corals are, well, they're wonderfully plastic. That's what makes them have such great evolutionary potential. But that also means they look, one species can look completely differently and have various, coloration patterns. So this is a huge problem. I've been trying really hard the last few years. We've got like a group of scientists from labs around the world, and we're trying to make a computer science challenge for this, where it can go out to the global CS community with a prize and the ego of who can crack this challenge. The hardest thing is just getting everyone to share their training data sets with their classifiers, because you need a lot of classifiers to develop accurate machine learning tools. And that's where, I don't know why people are so hesitant to do that. I mean, we're all in the same boat. We're all scientists. I think the more we support each other versus compete, the sooner we get to these useful tools. So I have all mine online. We're trying to get other people to put them online, just for that goal, is that it's time consuming now, but once we reach that threshold and we have large enough training data sets, we will eventually get there. Yeah, and then you can get to the level of doing huge spatial maps, because it wouldn't take you 5 years to annotate it, right? You could run it through these tools.
Awesome. Great.
Sorry, yeah, thank you for that answer. Great.
Next question is from Ryan Terrio. What is the process you use to photograph the coral reefs? What's the number of photos you take? How do you know when you have taken enough? What software do you use to process the photos? Have you used the structure for motion process that you use on coral on out of water objects?
Yeah, I 3D modeled my kids when they were babies, when they were sleeping, for fun. Yeah, you can 3D model anything. It works way better on land because you don't deal with the diffraction. So there's like, we use specific housings that are optimized to minimize distortion. But like I said, everything has gotten so good now that, I mean, I can send out students with very affordable cameras and you can still get great results. The software I primarily use is Agisoft. I've experimented with almost everyone out there. We also have Pix4D. The reason I use Agisoft, it's very sophisticated, works very well, and is the cheapest to license on multiple computers. That's the reality for that. For the photographs, we sort of you swim over the reef in like any kind of overlapping pattern. So you can go back and forth like a lawnmower, you can go in a circle, you can go zigzag. It just has to have enough overlap. So in like 100 square meter area, like 10 by 10 meters, you want 80% overlap forward and side to side. So that might take you about 1,000 to 1,500 images. And then you kind of go back and forth from there. Knowing when you have enough, it just takes time. The more you do it, the more you get a sense of your camera. Like I said, to Alex's question with the camera, the more you know and understand your camera, the easier and easier that gets.
Yeah. Okay, question from Paul Sporin. For the diver versus the lab comparisons, did the lab look at the 3D or the 2D images?
Yeah, great question. So we did look at the 2D mosaics, solely because those are mostly the most utilized products right now in the field. But we talked about that in the paper, that as people progress, you might get better results looking at the proper 3D models. So we do want to try the comparison with that as well.
Awesome. Okay, Monique Chiba has a question. The 3D reconstruction is very dependent on the collection of the 2D images. Drones do that very well, but it seems much more challenging underwater with divers. Also, to get accurate curvature, top-down pictures might not be enough. Finally, there are new techniques that do 3D reconstructions for 360 videos. That would save tremendous time, but not sure if that would work well in open space such as the ocean.
Yeah, so yeah, drones do well, true story. You can even use those on the ocean. Like, we do shallow sites at low tide with no wind, and you can map through the water. And Ved Chiriyeth at NASA developed a brilliant algorithm called fluid lensing. And so he actually removes liquid distortion from the water surface and
Oh,
can
wow.
map large reef areas, similar like Kaneohe. They tried it at Kaneohe. We tried to help them with that a while ago, but it's just so dirty, that bay. It was really hard to do. Yeah, and so, yeah, top downs work surprisingly well. The nice thing about it, there's no rule. So you can take oblique imagery, you can, as long as your images are overlapping, you can see a specific feature like a bommy and say, I'm gonna swim around that now and photograph it. You just need to keep the reef in your field of view. This is something I tell people all the time when they first start. Usually their biggest issue is they photograph too much of an angle and they get blue background. Now that blue background can't get matched to anything, it crashes the SIFT algorithm. So as long as it's the scene, you can get to any angle you want, but you would be amazed because at 24 millimeters, you actually, you look quite forward and back, even if you're pointing the camera down. So you capture a very, very good deal of side surface as well. With the 360 videos, we've tried it. We did make 3D models from GoPro Fusion. They sent us a few cameras to see if it would work. And we mapped underhanging areas and reef areas, but it is dramatically more distorted than anything you would use with a single lens camera. But yeah, you can do it.
Great. Okay, this is a question from John Bennett. Can the structure from motion method be performed using images captured from any type of camera? And are there any benefits to either use or compare these results to newly available LiDAR cameras? And then can you also repeat the programs you're using?
Yeah, absolutely. So you can use any camera. When I teach this in classes now, I have students download Trinio. Sometimes these apps come and go. Last time it was Trinio and I think Scans 3D or something, but there's apps even on your phone that can make 3D models just from pictures on your phone. You could use your iPhone and map it on the software. The software was Agisoft, AGISOFT. And then LiDAR is fantastic. I really like to compare our data to where we can get LiDAR data sets underwater. It's just hard underwater. LiDAR is very expensive to apply in an underwater environment.
Okay. Okay, next question is from Colin Anthony. Do you think we can use this concept on a smaller scale to reliably map surface area to track growth rate of more complex branching corals, or does distortion and complexity make it unreliable?
Yes, I learned a ton from archaeologists in Europe. They're really advanced in this field because they've got so many underwater cultural heritage sites that they want to virtually preserve. So I started going to conferences over there and just picking their brains, and they were extremely supportive and collaborative. And so they actually, one of those groups does this in Tahiti, and their whole entire goal is mapping like really slow incremental growth. And so in the short and dirty, it's possible. It's hard though when the growth is really slow, because if the coral, let's say, grows 1 millimeter and your error is about 1 millimeter, your now growth rate is within the bounds of your error. So if they're growing fast or there's a fast disturbance, the more that rate increases, the more confident you are in what you're measuring. But it is still hard with that distortion to capture very slow growth. And you have to be very, very fastidious. So you have to like have pins, you have to have ground control points. They always have to be in the same spot in order to accurately align them through time.
Okay, let's take two more. This is from Tom Blaney. John, have you been asked to do a talk for UH Maui College Sustainable Science Management Program? That sounds like an invitation.
Yeah, no, I haven't. So yeah, that'd be fun.
Okay, cool. And then one last one from Thomas Wolfgruber. His comment is Intel RealSense has some new and less than expensive 3D cameras in case you're interested.
Yeah, I've only heard about it. I still need to check it out, but I'm really excited for that. I think that will be a game changer where, you know, the next big step will be, you know, because it would always be better to have a true 3D camera or a true stereo camera, especially if you want to put it on autonomous vehicles, because you can't put out scale items and scale bars and ground truth points. So yeah, as those become cheaper, I think we'll probably go back to that and apply that for large area mapping.
Yeah, they're about 200 or 300 a piece. You just have to fit them into some sort of waterproof box or something. I imagine. Who knows? Needs a little testing.
Well, and that's where it gets funny because it's like with even the Fusion, the camera's cheaper than the housing. It's like harder to make the housing than it is to get the camera sometime. But yeah, I look forward. That'll be fun. I'll check that one out. It'd be fun to try test that.
Well, thank you, John. Everybody have a great weekend.